Dear students, we have studied the structure of blastocyst and we know that there are two types of cells in blastocyst. One is the outermost covering layer known as trophoblast layer and other is the inner cell mass layer or group of the cells which are constituting the inner cell mass. Now inner cell mass is going to develop into embryo proper, but the outermost layer which is known as the trophoblast of blastocyst, it is going to develop into four types of extra embryonic membranes. Names of the extra embryonic membranes are the chorion, which is going to be the outermost layer, then the amnion and allantois and yolk sac. These are the four extra embryonic membranes. Means in blastocyst, the trophoblast is not going to form any embryonic structure, but yes, it is going to help in development of embryo by formation of these four types of membranes: chorion, amnion, allantois and yolk sac. Now let's have a look how these membranes are formed around that membrane. Here in this structure, I have shown this embryo which is developing from the inner cell mass, and around this embryo you can see a number of structures. First of all, see that there is outermost layer. This outermost layer is known as the chorion layer or the chorion membrane is this. This chorion membrane is having finger-like structures. These finger-like structures are known as chorionic villi. These are actually those structures which will help in proper attachment of this whole structure to the wall of uterus or the endometrium. This villi will interdigitate in the wall of endometrium. Okay, so the endometrium will be attached with these villi, and these are those structures which will help in exchange of material with mother and the developing embryo or the fetus. So if we are going to talk about the role of this chorion, we can say that the chorion will help in attachment of the embryo to the wall of uterus by forming finger-like structures or projections, chorionic villi. It will also help in exchange of different types of materials through these villi. Okay, now come to the next layer. You can see here this red colored line. This red colored line is amnion, the second embryonic membrane, and you can see that between this amnion and this embryo, both are having a space in between, and this space is filled with a liquid here. This space is known as amniotic cavity, and this amniotic cavity is filled with a liquid known as amniotic fluid. Now what function this amniotic fluid or this amniotic cavity is going to perform? You can see that this embryo is floating in this liquid which is known as amniotic fluid. So this liquid is helping in protection of this embryo by absorption of shock. Means it helps as shock absorber. It will protect this embryo from any type of pressure change or any type of mechanical shock. And also it helps in regulation of the temperature around this embryo. It also protects this embryo from the desiccation conditions. Means if this liquid which is hydrating the conditions around this embryo, if this liquid is not present, then this embryo will dries up. Okay, so prevent its desiccation, it is necessary that this fluid should be around this embryo, and this liquid also help in movement of this developing embryo or the developing fetus. Movements of the embryo is possible just because of this liquid around this embryo, which is actually the cells of this second extra embryonic membrane, which we call as amnion. Okay, so these are the two types of extra embryonic membrane. One is the outermost chorion, second is the amnion. Now third type of extra embryonic membrane, you can see here, it is arising, this tubular structure is arising from the embryonic gut, and it is stalk-like structure. It is ending in a sac-like structure, bag-like structure. This sac-like structure is the third type of membrane, which is known as allantois. This is the allantois from which the blood vessels of the embryo will arise, and they will make a connection with that of endometrium through these blood vessels. And this stalk-like structure through which the blood vessels are running, this stalk-like structure is known as umbilical stalk. Okay, so this will help in making a vascular connection. This allantois will help in making a vascular or blood connection. Not direct connection is there. There are blood vessels only. Capillaries network will help in exchange of the various types of materials between the fetus and the mother. But yes, the blood vessels, they are contributed by this allantois. Now the fourth type of membrane, you can see here, it is a very small undeveloped sac-like structure, which is also linked to the which is present near the gut of this embryo. This is the yolk sac. Very small structure is there. This yolk sac means bag-like structure, but it is not having yolk, or it has very little yolk in it. It is actually, the yolk is actually a nutritive material which is necessary for those organisms whose development is external, in which the embryo will be developed on the cost of the food present in this sac. But you know that this whole structure in our case is going to attach to mother's wall, and we are going to derive the nutrition from the mother. So there is no need of any sac-like structure or bag-like structure that is going to provide nutrition to this embryo. Okay, so it is actually undeveloped. Yes, it is said that in initial phase of this embryonic development, this yolk cell help in formation of blood cells or the blood corpuscles. Okay, so this is the structure which is showing the location of all the four types of extra embryonic membranes. So let's recall, the extra embryonic membranes are the chorion, external layer, outermost layer having the villi, and the inner to chorion there is amnion, which is secreting a liquid, and that liquid will help in shock absorption. The third type of embryonic membrane is this allantois, which will help in formation of a vascular connection or establishing a vascular connection between the mother and fetus, while the fourth extra embryonic membrane is the yolk sac, which is actually undeveloped in our case. So I hope this lecture is clear to you.